All right, let's talk about something that sounds like it's straight out of science fiction. Regrowing human tooth enamel. It's the hardest substance in our entire body, and the big problem has always been that once it's gone for good. But what if that's all about to change? A huge new breakthrough suggests it just might.
Just let this number sink in for a second. Over 90% of adults have some kind of damage to their tooth enamel. We're talking cavities, general wear and tear, you name it. It's a problem that affects almost everyone, and our bodies just have no way of fixing it. So what's the deal? Why can't we fix it? Well, the reason is actually kind of simple, but also super frustrating. The special cells that make enamel, they're called ameloblasts, they do their job once, when we're kids, and then they disappear right after our adult teeth come in. It's like our body literally throws away the blueprint and the entire construction crew. So scientists realized, if we ever want to regenerate enamel, we first have to figure out how to build that crew from scratch.
So, if you want to build something new, what's the first thing you need? A blueprint, right? Or in this case, a map. And not just any map. They needed a super detailed cellular atlas to see exactly how a human tooth develops, right from the very beginning. And to do this, they studied human fetal tissue at all these critical moments in development. It was incredible. They were able to capture the entire journey, from the simple little thickening, through the bud and cap stages, all the way to this complex bell stage, which is where all the different cells get their final job assignments.
Now, the key piece of tech they use to do this is called single-cell sequencing. The best way to think about it is like creating a Google Maps for a developing tooth. It doesn't just show you the streets, it zooms all the way in and tells you what every single house is, who lives there, and what they're doing. It's an amazingly detailed census of every single cell.
And the result was this beautiful, complex map. It showed them 20 different cell populations, and this is the crucial part, how they all connect and talk to each other. This wasn't just about finding the famous ameloblasts, it was about discovering all their support cells, the whole ecosystem that you need to build a tooth correctly. Okay, so with this incredible map in hand, the next big challenge was to pull out the instructions. They needed to find the exact biological recipe for turning a plain old stem cell into one of these highly specialized enamel-making machines. You see, the map wasn't just a static picture. It actually showed them how the cells communicate with each other over time. They do this using things called signaling pathways, and you can think of these pathways as the step-by-step instructions in the recipe, telling cells exactly when to grow, where to go, and what to become. And this is where that recipe really comes to life. You can see how different signals fire up at very specific times during development. It's not just about throwing all the ingredients in a bowl at once. No, it's like a master chef knowing the precise moment to add each ingredient to get a perfect result. So, after all that work, they boiled it down to these 5 key signaling pathways. These are the core ingredients, the absolute essentials. And they even discovered that one of them, the WNT pathway, acts like a master regulator that kicks the whole process into gear. All right, this is where it gets really, really exciting. They had the map, they had the recipe. Now it was time to go into the lab and see if they could actually cook up some of these enamel-making cells for real. And the process was incredibly methodical. They started with human pluripotent stem cells, those amazing cells that can become basically any cell type in the body. Then, for 16 days, they bathed these cells in a perfectly timed cocktail of molecules, turning those key signaling pathways on and off, and guiding them step by step until, boom, they had early-stage ameloblasts right there in a petri dish. But how did they know they really did it, that they had succeeded? Well, they went right back to their original map. They compared the genetic signature of their lab-grown cells to the real fetal cells, and they found a 60% match. That's a huge deal. It confirmed that they were absolutely on the right track and had made cells that were very, very similar to the real thing. And this right here, this just shows how powerful their new system really is. They can now model genetic diseases right in the lab. For example, they grew this healthy, organized little tooth-like structure called an organoid. Then they knocked out a single critical gene. The result? A disorganized mess that perfectly recreated the cellular problems you see in a rare genetic disease called amelogenin imperfecta. This just opens up a whole new way to study these conditions. But here it is, the ultimate proof, the moment of truth. When they took their lab-grown cells and put them together with other dental cells, the ameloblasts not only got organized, they started doing their job. They actually secreted a hard,
mineralized enamel-like material. Wow. They didn't just look the part, they could actually do the work. So this is a massive scientific breakthrough, no question. But what does it actually mean for the rest of us? Let's take a look at the long-term possibilities and the future that this research is starting to build.
Okay, it's really important to manage our expectations here. This is not a treatment you're going to get at your dentist office next week. What happened in 2023 was the crucial foundational lab discovery. The next 5 years or so will be all about refining the technique to create bigger, more functional layers of enamel. But the long-term goal, that's developing real cell-based therapies to actually repair our teeth.
So, why does all this matter so much? Because the potential applications could revolutionize dentistry. I mean, imagine being able to treat those genetic enamel disorders for real. Or think about living fillings that actually merge with your tooth to repair a cavity from the inside out. We could even be talking about regenerating tooth structure that was lost in an accident.
And don't just take my word for it. The researchers themselves said it best in their paper. This work paves the way for future regenerative dentistry. This is a fundamental shift in how we think about dental health. We're moving away from just patching up damage and towards truly regenerating what was lost.
And that leaves us with this one truly fascinating question. We've always just accepted that we get one set of adult teeth, and that's it. Game over. But by decoding the fundamental biology of how a tooth is built from the ground up, have these scientists just unlocked the very first step toward a future where we might one day be able to regrow our own? Just think about that.